Welcome to my channel Future Academics. In this video today we will discuss primer design, which is a fundamental topic in molecular biology. Primers are essential in techniques involving DNA synthesis and hybridization, and therefore understanding how they are designed is key to successful experiments. So let's get started. Primers are also known as oligonucleotide or simply oligos. They are short single-stranded DNA molecules typically ranging from 6 to 22 nucleotides in length. Sometimes the size or the length may be more than 22 nucleotides depending on your needs and experimental design. In this target DNA, the red sequences are the putative selected site for primer. Therefore, the primer must be complementary to allow it to bind precisely to their respective sites. For selection of primers, primers are typically designed using the published cDNA sequence as shown here, which mirrors the messenger RNA coding sequence with thymine replacing uracil. The first and foremost critical step in primer designing is to select an appropriate region from this cDNA sequence for designing the primer. For the hybridization and sequencing applications, a single complementary primer is sufficient. However, for PCR, RT-PCR, two primers are required, one complementary to each strand of the target DNA. So selection of the PCR primers, the first thing, follow step by step. Select a region of consisting of 150 to 350 bases long region from the target DNA for PCR amplification. Select upstream primer toward the 5' end, which is shown here, upstream, and downstream primer toward the 3' end. The GC content of these primers should be approximately 50% each, and their TM should match also. So selection of the upstream primer for PCR. Let us design the upstream primer or the sense primer for the selected highlighted region here, this one. The upstream primer is selected toward the 5' end. It consists of 15 to 22 nucleotides from the 5' end, and this must bind to the complementary sequence of this target. Therefore, copy the sequence and define the 5' and 3' ends of the sequence, and your primer is ready for your PCR reaction. So let us design the downstream primer for amplifying this highlighted sequence here. Downstream primer is also known as antisense primer, or antisense means complementary to the selected region. The primer must bind to the site shown here. Therefore, copy the sequence and reverse complement using any bioinformatic tools available online, and define the end 5' and 3' end, and your primer is ready for your PCR reaction. For a successful outcome and successful experimentation, the following criteria should be observed while designing the primer. The first one is select a 15 to 22 numeric primer sequence having approximately 50% GC content. The TM of these primers should be high. TM can be calculated using this formula. TM is equal to 2 into number of A and T plus 4 times number of C and G together. That for PCR reaction, it is important that the TM of both primers, up and downstream primers, should be the same. It is also important to avoid cross reaction. Therefore, blast your primer sequence and select the primer avoiding cross reaction. So thank you very much for watching this video, and I hope that you understand the concept behind designing primer. Regarding the application of these primers, I have uploaded the individual or dedicated videos on PCR and RT-PCR as well as DNA sequencing, so you can approach these on my YouTube videos. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.